right. Hey, y'all. This is our section. That's really not necessarily a section in your book. The food tests are actually embedded into each area for carbohydrates, protein, fat, and of course, DNA. But the way that I thought would be easiest this year, trying it like a little differently, is not to give you so much information overload all at once and kind of divide it. So I'm doing the food test section really as its own standalone. So this is a very important table, and you actually have this table for your notes today. This table gives you every single food test that you will be responsible for knowing for your end of the year Cambridge exam. It's going to be really important to write down everything in this table and really get familiar with it. And then we're going to watch videos for each of these labs because there's a lot of moving parts and it's really hard to get in our lab right now with everything going on. So let's look at each food test. The first is for reducing sugar. The first two are actually for carbohydrates. Reducing sugar is going to be any type of sugar that is either a disaccharide, typically a maltose or a sucrose, that can be reduced down to its glucose form, and we can test for that. We test for that using Benedict's solution. So Benedict, what we are going to do for our method is add Benedict's solution to the test tube with food. We heat it in a water bath, and then we observe. Our start color will be blue. The end color for a positive result, brick red. The test for starch, fairly simple. It's using iodine solution. We will add a few drops of the iodine solution straight to the food. The iodine begins as a brown orangey color. And then if starch is present in that food, the resulting color will be a blue-black, a very dark color. And then we move on to protein. A protein, to test for that, we use Burette's reagent, or just you can simply say we use Burette's. We will add the Burette solution to the test tube with the food, shake it very gently. Once you do that, you will see that the Burette's begins as a start color of blue, but the end color, if protein is present, will be purple. And then moving on to testing for fats. For a fat test, we call this the emulsion test. An emulsion is this cloudy look that you will be able to see in the video for this test. We will dissolve the food in ethanol, pour the solution into a clean test tube, and then simply add water. Once you see this, the beginning color will be clear. And then once we add in that water, a white emulsion will appear. Okay, and then our final test is for vitamin C. This is the DCPIP, or you might hear it phrased as the DCPIP test. We will add DCPIP to a test tube with a food sample. The solution will turn clear if vitamin C is present. So it begins as a blue or a dark purple. It's very, very, very dark. And then clear, the color that is very clear will be present, or it will turn to a clear color if vitamin C is present, but it will stay blue if it's not present. You will also be able to see that on the video. So make sure you get this table in your notes so you can check back in with me with your CFU the next time we see each other.